Good morning, everyone. Welcome to another Terasaki seminar series. Today, we have Professor Zorlutuna from Notre Dame University. She is the Sheehan Family Collegiate Professor of Engineering in Aerospace and Mechanical Engineering Department, Chemical and Biomolecular Engineering Department, and in Bioengineering Graduate Program at the University of Notre Dame. She currently serves as the Acting Associate Dean for Diversity and Faculty Development at Notre Dame College of Engineering. Her research explores designing biomimetic environments for understanding and controlling cell behavior, cell-cell, and cell-environment interactions using tissue engineering and micro and nanofabrication approaches. She has received numerous awards, including NSF Career Award and PKs, and she's a National Academy of Sciences Kavli Fellow. The floor is yours, Professor Zorlutuna.
Thank you so much, Dr. Dökmeci. First of all, I would like to start by thanking Professor Kadam Hüseyni and Mehmet Dökmeci for inviting me to give this talk today. And as one of the alumni of Kadam Hüseyni Lab, this is like homecoming. I'm very happy to share my latest research with the audience of Terasaki Institute. And today I'm going to talk about tissue-engineered disease models to study the effect of age on disease progression. But I would like to start by giving an overview of my lab. So we have two research trusts, and the first one is on cell-based robotics and computing. I will briefly introduce that. The second trust is on engineered disease models, which will be the topic of this talk. About 30% of my lab works on leveraging cardiomyocytes and their interactions with cardiac fibroblasts for exploring cardiac-based devices, microfabricated cardiac tissue structures for biocomputing and biorobotics applications. And we have explored using the native properties of cardiac muscle cells and fibroblasts and their directional signal transduction in the heart by patterning them in certain microfabricated structures, and looked into possibilities of using them as logic gate components such as diodes, AND gates, and OR gates. And we have also looked into their coupling dynamics. So cardiac cells beat spontaneously, and once they are provided a certain coupling measure, such as a fibroblast bridge, they would couple to a collective frequency, and this can be mapped using microfabrication methods to solve computationally hard problems. And working with computer science and electrical engineering collaborators, we have developed a system where we use these beating heart muscle cells as biooscillators and explore the coupling dynamics of micropatterned biooscillators for solving computationally hard problems such as graph coloring. And this is significant because although biocomputing is an attractive field within the bioengineering and biotechnology, cell level systems have yet to be explored more. Most of the systems that are using biocomputing to solve computational problems look into subcellular dynamics of DNA and protein pathways, and those pathways are slow in nature, as opposed to heart cell beating, which is much faster. So actually the platform that we are developing can possibly give a much faster and energy efficient alternative to be used in biocomputing applications. And so far we just it is in press, so it will be published very soon in Advanced Intelligent Systems. We have developed a 64-node biooscillator system where we were able to solve a computational hard problem, the graph coloring problem, faster than the current algorithms, such as Johnson algorithm. And we also work on developing technologies, micro and nanoscale. In this specific example here, it's a nanophotonic potential sensor for being able to better track the cell membrane potential changes and use that for either biocomputing or biorobotic applications, as well as in biomedical applications, where you can look into changes in subcellular level, changes in cell membrane potential, and therefore look into cell coupling dynamics, which can either be used for the applications I just mentioned or in applications in biomedicine, such as arrhythmias. And these biocomputing platforms either can be standalone computing devices, which are energy efficient and possibly integratable with living tissue, as well as they can be parts of biorobotic devices, such as the one I'm showing here that is a floatable device, which is propelled by the beating activities of the muscle cells in the tail here. So as you can see, we are very invested in cardiomyocyte mechanobiology, and micropatterning. And with that, it comes also the tissue engineering perspective of cardiac tissue. And in the other trust, we are very interested in looking into organ on chip and tissue engineered disease models for studying heart disease, as well as some cancers. And we are specifically interested in looking into cell extracellular matrix interactions using these organ-on-chip platforms, where we look into the microfabricated ECM interactions with the cells for either studying disease progression and initiation or stem cell differentiation and function. One particular aspect of this broader area that we are focused on is the age of the extracellular matrix. And in our engineered disease models, we oftentimes try to replicate the native tissue environment with respect to its aged context, and changes that are occurring with age is oftentimes our goal in creating the tissues that we are developing for studying heart disease or cancer. We are also interested in using these organ-on-chip devices to study organ preservation and biomarker discovery. So in today's talk, I will be mostly focusing on cell-ECM interactions and our systems that we developed to study the biology that is changing the ECM, as well as in turn that ECM's phenotypical changes that caused on the cells that are seated on that ECM. So just to motivate this broader area of engineered disease models, I would like to highlight this figure from a study that was published several years ago in Nature, where they compared the gene expression patterns of specific organs from human and mouse. And as you can see in this figure, the human organs tend to correlate very well and cluster on one side of this graph, while the mouse organs cluster together. And that alludes to the fact that human and mouse organs differ significantly, and to a degree that if you look at these squares that are filled, which is human organs, you see that in human intestine and human kidney are more closely correlated than human intestine and mouse intestine, which has no correlation at all. And if you look at the cell level and organelle level gene expression patterns, you'll see that there is a certain degree of obviously evolutionary conservation of subcellular systems like nuclear, intercellular, and organelle level gene expressions and pathways, while cell-cell and cell-ECM interactions like vasculature, motility, immune system cell adhesion, and signal receptor transducer pathways, they are not much correlated in between mouse and human. And this is why there has been a huge effort in the past decade or so for using human relevant disease models and engineered tissue models for complementing preclinical research and trying to remedy these discrepancies between mouse and human physiology. And these cell-cell and cell-ECM interactions oftentimes are at the core of disease initiation, disease onset, and progression. So in this talk, I will be giving an outline of our efforts in tissue-engineered disease models in part 1 for breast cancer and in part 2 for myocardial infarction. And specifically, I will focus on the role of aging extracellular matrix in both. And also in both, I will talk about extracellular vesicles and how we can leverage the extracellular vesicle presence either in the matrix itself or in a heart-on-a-chip device in the case of the myocardial infarction study for understanding better the disease progression in these tissues. So cancer incidence increases by age, and it is one of the single most relevant factor that correlates with increase in cancer incidences. So far, the reasoning have been sought in cell level changes with age, like telomere shortening, accumulation of mutations, impaired repair mechanisms, and impaired immune surveillance. And studies focused on aging-related alterations are mostly studied in cells. And although there is no doubt that such changes contribute to the increased incidences of cancer with age, extracellular matrix itself changes by age, and this has been less explored. And our goal here was to study the changes in breast cancer, healthy breast tissue ECM by age, and how those aging ECM factors may be contributing to increased cancer incidence by age. And I will talk about 2 different studies. One is published last year, which is the changes in extracellular matrix of healthy tissue, and then one about the matrix-bound vesicles that we have identified in breast ECM and how they change by age. And the second study is about to be published and not online yet. So it is known that extracellular matrix is a dynamic structure, and it changes in development and age. And among the things that change during the ECM aging is biochemical composition, stiffness, pore size, porosity, organization, and alignment. And although these factors have been studied here and there separately, there has not been a comprehensive study to look into all the changes happening by age, and no study to see how that aging ECM may be supporting cancerous phenotype. So our hypothesis here was to study the aging ECM, and then the hypothesis was the aging ECM supports cancerous phenotype. So this figure summarizes the significant changes we have seen in the healthy breast tissue with age. And this figure here shows our studies with the mouse tissue, but we have repeated and complemented all these points that we have seen changing by age with human tissues too, and I will show some data on that, but that's unpublished and is in preparation for publication. So it will come out later this year, but all these changes that I'm gonna outline here that we studied using mouse tissue pretty much correlated with healthy human breast tissue too. So we have decellularized the native tissue, and then because breast tissue has a high degree of fat, we have also delipidized the tissue. And then we looked into fiber structure in terms of its alignment, diameter, and also the mechanical properties of the extracellular matrix after decellularization as well as delipidization. And we have seen that the young matrix has thicker bundles of collagen and they're straighter, while the aged one shows curlier and thinner but more dense collagen structure. This shows the STM images, and later on in the next slide I have a second harmonic showing the collagen structure as well. And then the fiber diameter significantly decreases by age in both the native decell and the lipidized tissues. When you look at the mechanical properties, although the overall stiffness of the tissue seems to be not increasing that much as you age. If you delipidize the tissue and get rid of the lipid components, the aged tissue has significantly stiffer matrix than the young one. So if you look at the native form, you see the young and aged almost similar. Maybe aged is slightly softer, but this softness during the non-indentation comes mostly from the fact that there's lipid there. And then as soon as you start getting rid of the lipid, you see that increase in the stiffness. So the collagen itself is denser and stiffer. And if you look at the content of this extracellular matrix in terms of the cytokines, you see that the pro-inflammatory cytokines that could potentially favor EMT and metastasis are enriched in the aged tissue in both the native form and decell delipidized form. And in terms of the macromolecular structure, there's lower collagen 5 and collagen 15, as well as lower lumicin by age. And you have slightly higher fibrillin, but definitely higher collagen 1 by age proportionally, so as we assessed by mass spec. And so after observing all these changes by age in the extracellular matrix, what we wanted to see is how this decellularized and delipidized extracellular matrix may be affecting the cell phenotype. So we started by just a cancer cell line, which is MDIMB231. And these videos that I'm going to show you shows the collagen structure, which is shown with red fluorescence. This is second harmonic imaging, so there's no staining here. And then the cells are tagged with GFP. So the green cells that are moving around these decellularized matrices are co-imaged by second harmonic and confocal. And as you can see from the videos, the cells that are seated on the age matrix moves much faster, and also they divide more compared to the ones seated on the young matrix. And if you look into the quantification of this, the motility is more than double on the aged tissue compared to the young. This is for MDA cells. And because the cancer cells acted so drastically different, we were curious to see what normal cells would do. This graph on the left marked KTB21 cells are just healthy normal epithelial cells. They also increase their motility by twofold. And overall, of course, the cancer cell is faster compared to the motility motility of the normal cells if you look at the absolute values of the motility. But in both cases, we saw that cell motility almost doubled. And when we did a invasion assay with these cells using a transfer plate, the amount of cells that invaded was also doubled when the cells were seated on healthy aged tissue compared to healthy young. And this was true for normal healthy epithelial cells too, which was very interesting for us. And we wanted to further explore what is changing in these normal epithelial breast cells from healthy donors that this age matrix is doing to them. So first of all, we wanted to study the phenotypical changes because these are healthy epithelial cells. When you see these on decellularized and delipidized matrices, they form these spheres, which is an indicator of healthy epithelial biology structure. When you look into ECAD expression, the ECAD is in the cell periphery, and cells are nicely intactly contacting each other and forming these structures. Same cells on the age matrix form those similar spheroids, but the spheres start to deform after a week. And then by day 10, they are considerably losing their roundness. And by day 12 and beyond, they lose their integrity. So the spheres cannot stay intact for these cells that are seated on age matrices. And when you look into the ECAD expression, the cells lose their peripheral ECAD, and then ECAD is accumulated mostly in the nuclear region. So we were curious to see what is causing this in terms of the changes in the phenotype of these healthy epithelial cells, and we have identified after single cell transcriptomic analyses 29 genes that were upregulated in these normal cells just because they were seated on these aged matrices. And we identified different clusters, 9 of them, and cluster 7 was enriched on aged cells that are seated on aged matrices, significantly more than the cells seated on young matrices. In other words, there's a subpopulation of these normal cells that are otherwise identical. Once they're seated on the aged matrix, a subpopulation of these cells started to enrich in terms of their upregulation in genes that are specifically identified by EMT and invasiveness. And we have seen increased cell migration and invasion. Some of these genes are listed here. This is the cluster 7, and many of these genes that we identified that are highly differentially expressed in the cells on age were correlated with breast cancer. And again, this is the motility and invasion data that I showed in the previous slide that is showing this drastic change in cell motility and invasion. So one of the genes that we identified and have seen that it is significantly upregulated was lysyl oxidase. So we wanted to see what happens if we were to knock down lysyl oxidase by siRNA treatment. So what we did is we seeded the cells as we did in the earlier study on aged and young, and then we treated the cells either with scramble siRNA as control or LOX siRNA. And when we treated the cells with LOX siRNA, interestingly, we were able to rescue the phenotype. So in this, in the cells that we just had the control SI scramble RNA, the cells couldn't keep their intact sphere phenotype, and the spheres dissolved as we have observed before, while the cells, despite being in the age matrix, when we treated with lysyl oxidase siRNA, they were able to keep their phenotype in the age matrices as they did in the young. So the left is the young scramble and LOX siRNA, and right in the is the aged and the age matrices with scramble, where you don't see any of the spheres, and the lysyl oxidase, the siRNA, kept the spheres intact. Not only that, also in the case of the cells that are treated with lysyl oxidase siRNA, despite they are being on age matrix, they were able to keep their E-cadherin localization to their membrane. And then we were able to reverse the phenotype of increased migratory behavior just by treating with lysyl oxidase siRNA. So again, this is the quantification of the normal epithelial cell trajectories in the young matrix and in the age matrix. The LOX siRNA was able to reverse the phenotype that we have seen, along with some changes, reversal of the MMP expression and ECAD localization. So what we are currently doing in this study, after validating these results with human decellularized and delipidized ECM, we are in the process of implanting these cell-seeded matrices, this time seeding them with cancer progenitor cells like DCIS-COM cells, and cells that can initiate cancer in immunodeficient mice. In this case, we are using a REG1 negative mouse model. And when we implanted the young and the aged matrices that contain cancer cells, and same amount of cancer cells, same mouse, one side is the young and the other side is the left, we have seen 80% larger tumors formed just caused by the presence of the aged matrix that was implanted along with these cells. So now we are in the phase of repeating these experiments with the human tissue, and we are looking to understand what in the age matrix may be causing these changes, gene changes that we have identified. What is the role of lysyl oxidase in the case of cell phenotype change and the transition to a more EMT-like phenotype in normal epithelial cells? And then what can be leveraged in this to understand better the causes of increased occurrence of breast cancer by age from an ECM perspective. And one thing that we identified that is significantly changing by age is the matrix-bound extracellular vesicles that are stocked and accumulated in extracellular matrix. So extracellular vesicles, or EVs, are small membrane-derived particles that are sent out from the cells, and they are crucial in many cellular processes, especially in terms of cell-cell interactions, and they have been gaining interest in the past several years. But more recently, us and others also identified extracellular matrix-bound extracellular vesicles, and some of them are exosomes, and some of them are larger extracellular vesicles, but we are specifically interested in the ones that we stumbled upon in the aged breast matrix. And I will talk about the corresponding study that we are doing in parallel with the ones that we identified in the heart matrix. But this is very exciting because this is something really new, and this can be the ECM-bound extracellular vesicles can be a major player in the ECM effects that we have been observing for past several decades. And so we use a very mild decellularization protocol that doesn't harm the EVs, but still get rid of the cells and cellular content. And then we follow a protocol for extracting these extracellular vesicles. And when you look at the NTA nanoparticle tracking analysis, you see the size is right in the range of exosomes, which is slightly shy of 200 microns. And we look into the differences in aged versus young tissue bound membrane-bound vesicles. They are positive for canonical exosome markers, CD9 and CD63 in this case. This is immunogold assay and TEM for identifying the structure of these extracellular vesicles. I would dare to call them exosomes because they are like CD9 and 63 positive and in the right size. And we were able to identify them and extract them both from aged and the young tissue. And here in this TM, you can see them in the intact native tissue. And this on the right shows the isolated ones with the markers shown. So then what we were interested to see was how the cells would respond to these extracted EVs from the decellularized tissues. So we kind of repeated the experiments we have done in the previous study, but this time instead of seeding the cells in these decell and the lipidized tissues, we have treated cells seeded in culture well plates with the extracellular vesicles that we have isolated from aged and young matrices. And then we have done this with the cancer cells, which is on the left, MDA cells, and then normal cells, normal epithelial cells, which is the KTV cells on the right. And this figure shows the uptake. So both cells do uptake, both the young and the aged vesicles similarly. The cancer cells uptake them faster, which is in agreement with the observations in the literature. And obviously, interactions of EVs and the exosomes and their involvement in cancer have been explored a lot. But so far, researchers have been focused on the EVs or exosomes from biofluids or cell culture supernatants. So the difference here is the main difference here is these EVs are extracted from extracellular matrices, and they are bound and stuck in the EVs in the ECM otherwise. So when you treat these cells with the EVs that we extracted from the ECM, we also shown a significant change in their motility as well as their invasion. And then the age also was a big factor. Here on the left in both C and D, part C and D, you see the cancer cell motility and invasion. And then on the right, you have the normal epithelial cell motility and invasion. In both cases, the treatment with matrix-bound vesicles promoted cells to become more motile and invasive. And we also wanted to see how the cells may be able to pick these EVs as they go through a 3D ECM-like environment, because the study I show on the left is just cells seated on transval plates and treated with these extracellular vesicles that they're uptaking. So what we wanted to see is if these vesicles are actually stuck in the ECM, would the cells be able to engulf them as they go through a 3D matrix? So what we did is we have embedded these extracellular vesicles in a 3D collagen matrix, and we have tried to see if the cells would invade more. And we tried it both ways. In one case, we treated the cells, pre-treated them with the matrix-bound vesicles, and then seated them on top of a collagen gel that we inserted in a transval plate, and looked into the number of cells that are invading. So this was to see how the pre-treated or primed cells, how their 3D invasiveness is changing. And the aged matrix-bound vesicles cause the cells to invade through the collagen and the transval plate more. But more strikingly, when we encapsulated these extracellular vesicles within collagen gel, along with cells were able to uptake. And the images here shows the cells only, and then cells with after treating or waiting for the cells to go through, migrate through a gel with Exaglow labeled extracellular vesicles, and you see the orange cells. So cells, as they migrate through the collagen gel, they pick up these EVs, and then they end up going other side of the transval plate. And as they go through this process, the ones that capture the aged EVs end up becoming faster, and more cells invade quicker through the 3D collagen environment. So obviously, we were interested in what may be the cause of this. So we studied the content of these vesicles. We have looked into cytokine content too. In terms of cytokines, we have seen the most drastic change in adipokine content. The aged vesicles had a significantly more adipokine content than the young ones, but majority of the other cytokines was not drastically changing. Again, keep in mind that these are vesicles that we extracted from healthy tissue. This is the only difference is the age of the tissue. But when you look into the microRNA content, which is gaining more and more interest in the context of extracellular vesicles, the aged tissue had a huge amount of microRNAs that are significantly upregulated compared to the young one. And when you look at the most highly upregulated ones, you see that several of these are associated already in literature from individual studies with cancer. So this is like microRNA 10B, 30E, and 210. Very significantly in this, on the right panel, you see the PCR validation of the nanostring analyses we have done with these microRNA panel of 800 microRNAs. And these three we validated with PCR that are already shown to be positively correlated with breast cancer occurrence, was highly significantly expressed in the matrix-bound vesicles that we isolated from the aged matrix. And what we did next is we treated these cells with the mimics of these microRNAs directly instead of treating with the vesicles. And we have seen an increase in the invaded cell count, both in the case of cancer and the normal epithelial cells when we treat these individually, as well as when we treat them as a cocktail. So individual microRNAs that were highly significantly expressed in the aged extracellular vesicles that are bound in the matrix cause the cells to invade faster and more compared to the negative control. But when we treated them as a cocktail, there was a synergistic activity and the cells invaded even more with the treatment of the cocktail. So to summarize this part, on the aged ECM, the deformation and disruption of normal epithelial spheroids caused by E-cadherin delocalization and increased MMP2 levels was a major observation that can potentially explain the increased enhanced occurrence of breast cancer by age. And when you look into gene level changes in these cells, we identified several genes that are highly upregulated, and at least one of them, lysyl oxidase, blocking it, reversed the phenotype in the cells that are seated on the aged tissue. And we were able to reduce the cell motility and invasion that caused we blocked the increase in cell motility and invasion that was caused by the aged extracellular matrix. And there needs to be further assessment of what is causing these changes. Also, what's the contribution of decreased collagen 5, 15, and there are other increased chemokines and cytokines. But overall, the overall changes in the matrix cause this phenotypical changes in normal epithelial cells, and identifying these would significantly help to target new therapeutics in prevention of breast cancer occurrence in aged population. And among the changes that are happening in the aged tissue, one novel thing we stumbled upon and started to investigate was the matrix-bound vesicles that we identified in the breast ECM. And they are bioactive, and they can affect the breast cancer progression and invasion. And the ones that we identified in the aged matrix definitely promoted more cancer-like behavior in both in normal and cancerous cells. And we have identified several microRNAs that may be the reason for the observed phenotypical changes. So in part 2, I would like to give a parallel story from a heart disease perspective. So similar to the cancer occurrence by age, heart disease occurs more in elderly individuals. And then again, aging is one of the biggest risk factors for heart disease. So myocardial infarction not only happens more in the older population, but also when it happens, the damage in the muscle is much larger in the older population. So we performed parallel studies to what's changing in the aged heart muscle and how it may be contributing to the increased infarct size in the older population. And I'm going to talk about specifically, in the interest of time, the extracellular vesicles we identified in the heart tissue. And this study is under review right now, under revision actually, but the full story, you can find it in bioRxiv. Here is the link. And we specifically wanted to see what may be different in terms of the extracellular vesicle content of 4 different cohorts. And in this study, we directly looked into human heart tissue. So all the results I will show here is from young and aged male and female heart tissues that we collected from Indiana Donor Network. These tissues couldn't make it for transplantation, but they were not discarded based on the heart tissue function. So these are as healthy as you can get in terms of a human heart tissue. And there were other things that were going on with the patient that deemed these hearts not appropriate for transplantation, but otherwise they were, in terms of the muscle tissue itself, as healthy as you can get from a human perspective. So we have identified these extracellular vesicles in all 4 cohorts. And interestingly, this is not something we have seen in the breast tissue bound vesicles. In the heart tissue bound vesicles, we have observed smaller vesicles in the aged tissue. Both aged male and female tissue had significantly smaller vesicles compared to young male and female tissue. Probably because of this difference in the size of the vesicles that we isolated from the aged female and male were uptaken much faster by the cells. So in this particular study, we are looking into how these extracellular vesicles that are isolated from 4 different cohorts we had is changing the phenotype of human fibroblasts. Our working hypothesis is the extracellular vesicles that are stuck in ECM modulates the fibroblastic response upon a myocardial infarction and their transition to myofibroblastic lineage. And for that, we first started by studying the uptake. All the vesicles were uptaken to a large degree. Again, the ones from the aged tissue were uptaken faster. And when we seeded these cells in collagen gels that were treated with 4 different cohorts of ECM-bound extracellular vesicles, the fibroblasts contracted the gels differentially. And I'm going to go through the results, but we did 2 different studies, treated the cardiac fibroblasts, human cardiac fibroblasts, with extracellular vesicles from young and aged male and females that we isolated, and then we performed either wound healing, which is in the form of a scratch assay, or this is a 2D assay where we are looking into closure of a wound, and then we looked into the contraction of the collagen gel that are populated with 3D encapsulated human cardiac fibroblasts that are again treated with 4 different types of ECM-bound vesicles. And then we also looked into the alpha SMA production of the cells, and alpha SMA production is an indicator of myofibroblastic differentiation, which we want to avoid for a healthy myocardial repair upon myocardial infarction. So first of all, our control was cells that are treated with TGF-beta, and we had also controls where we didn't have any sort of treatment. In the case of the aged male extracellular matrix bound vesicles, the closure was the least. So the cells that are treated by the vesicles from aged male ended up not closing as much as the ones that are treated with young or female groups. Similarly, looking into contraction of the collagen gel, the aged male group contracted the collagen much more than TGF-beta treated cells. And then we had the female group responding better in terms of the contraction of the collagen gel. And when you look at the alpha SMA production, a young female group ended up having the least amount of myofibroblastic cells, while the aged male had even more than the control group. So when you look into the content of these different left ventricle extracellular matrix derived vesicles, there's a huge increase in inflammatory and aging related cytokines, which is oftentimes referred to inflammaging, in aged males. And this is kind of pathway analyses that shows the differential expression of different pathways that are relevant to myocardial infarction and the subsequent healing. And in all cases, the aged male ended up giving the worst pathway profile in terms of healing potential. And we again looked into microRNA profile, and we have seen a huge difference in expression of the microRNA content of these vesicles from different cohorts. And for the interest of time, I will just focus on the 4 that we have identified that is significantly differentially expressed in the young and age groups, as well as female and male groups. And we targeted these 4 that we have validated differential expression of, which we have seen with nanostring analysis. We have validated with PCR, so microRNA 125, 143, 199A, and 222 is differentially expressed, and especially found very little in the aged male group, while significantly higher in the female and or young group. And we again did a similar study where we added mimics of these microRNAs in the culture, where we have the cardiac fibroblasts, and repeated our wound closure and alpha SMA expression studies. And we have seen similar trends when we treated the cells with microRNA cocktail, like the results we have got with young and female groups compared to the aged male. Interestingly, not only the wound closure and myofibroblastic differentiation was lower upon treatment with this cocktail and upon treatment with young and female EVs, but it was also interesting to see that the cell death was much less. So this is an anoxia treatment post myocardial infarct study where we treated the cells with no glucose, no oxygen condition for 3 hours. And we saw that the cell survival was much better when we added this microRNA cocktail. And so this kind of shows the importance of the tissue resident extracellular vesicles. And obviously we are an organ-on-a-chip lab, so we want to leverage the organ-on-chip technology, specifically heart-on-chip technology we have developed here. I'm showing you a snapshot of the most up-to-date device we have. We have these cross-channel device where we see iPSC-derived cardiomyocytes after maturating them using various different fatty acid cocktails, and ensuring that their gene expression specific to cardiac markers is as close to as left ventricle as possible, which is shown by this heat map on the left. Then we see the device with endothelial cells that we also differentiate from iPSCs, and then we look into the cell expression of extracellular vesicles. And this is just to establish the baseline. We can kill the cells to a certain extent that is similar to the heart attack patients. And then when we look into extracellular vesicle production of these cells and compare it with the plasma samples that we obtain from post and pre-myocardial infarction patients, we were able to mimic a similar extracellular vesicle production using this heart-on-a-chip model compared to the human plasma. And we were able to integrate this organ-on-chip device with a sensor that rapidly senses microRNA instead of PCR. So this is a membrane-based sensor that has an onboard exosome lysing unit, which is ultrasound-based. So as the media flows through the organ-on-chip to the sensor, it sits like for 20 seconds on this sonic acoustic wave device where the exosomes are lysed, and then the content of the exosomes can be measured with the membrane sensor downstream. And we were able to show that the heart-on-chip device produces similar kind of exosomes as shown here on the right with the Western blot. They have similar surface markers and lacks the cell markers, indicating that these particles we detect are not cell pieces but genuine exosomes. And then the microRNA content as shown here for 4 different microRNAs is similar to the plasma samples. With that, I want to summarize part 2, saying that functional exosomes are existing in the extracellular matrix of left ventricle tissue. And given that traditionally exosomes are isolated and examined from biofluids, it is important to study and understand these ECM-bound vesicles, and especially how drastically they change in terms of their cargo content by sex and age, can lead to studies where one can identify targets that are highly expressed in populations that less severely experience a certain disease. In this specific case, young females rarely experience heart attack or adverse consequences from it. So looking into what kind of cues there exist in the young female tissue as opposed to aged male helped us to identify this specific cocktail, but there are so many other differentially expressed cargo content of these extracellular vesicles that is probably there are like hundreds of more targets that can be explored there. And a heart on a chip device that we have studied shows similar exosome and microRNA expression profile to the patient profiles that we obtained from clinical samples, and it can be used to either identify biomarkers or study the origin of biofluid-bound or tissue-bound extracellular vesicles. With that, I would like to thank to my lab, my funding, and my collaborators, and happy to answer any questions you may have. Thank you.
Hi, Pinar. Thank you for the exciting talk. Several questions came in. So one of them is, cancer cells produce a significant amount of ECM. Also, the production of ECM decreases with age. I'm curious to know if you already have information about the level of ECM in aged model cancer versus the young one.
Great question. So all studies we have performed with young and aged tissue are from healthy samples. We have not looked into young versus aged cancer ECM, but it's something that would be very interesting to look into and study. We are more interested in the healthy tissue and what changes in healthy tissue by age, with the hopes that we can come up with things to prevent the occurrence of cancer by age. So that's why we are focusing in the healthy tissue.
Thank you. Another person is asking about the decellularization protocol. He thinks that you're using 0.1% PAA, 4% ethanol. For how long the tissue is treated with PAA, and did you quantify the DNA content?
Yeah, we have quantified the DNA content that's below the limit that you can call your tissue decellularized. The duration of treatment, you may need to ask it to my graduate student. So I don't remember on top of my head because I'm not the person obviously doing the experiments in the lab. But if that person who is asking the question wouldn't mind emailing me, I would be happy to share the protocol. I just don't want to say something wrong and I think the protocol is the bioarchive link I gave. I can go back to that slide. The protocol is published online. This link here will have the protocol in detail if anyone wants to look into that.
Okay, I told the guy to email you. A couple of things about the interactions of EVs and cells. How or why do they get into cells and interact?
So EVs are a way of communication in between cells, and the reason usually is to regulate hemostasis in the native tissue, but like in most hemostatic cases that changes with age, malfunctioning would cause disease initiation and progression, but it is a natural way of cells talking with each other. Why they end up getting stuck in extracellular matrix is something we are looking right now.
Thank you. Another question is, you mentioned an inline detection exosome lysing chip with a membrane sensor. Is it single use or multi-use? Can you regenerate the surfaces?
I believe it is this one. So we use it for single use, but I believe you can possibly, and it is so cheap, these membranes you can buy bulk in meters. So the more expensive part is functionalization of it. So we never try to reuse, but I believe it may be possible if you wash it, you can refunctionalize it.
Thank you. One other question is, you're doing great work in organs on a chip. Where do you see the field moving? Do you see some immune activity or complicated fluidics or IPS, or where do you see this organs on? I mean, it's been come a long way for 10, 15 years. Where do you see the field moving?
Yeah, I think immune interactions for sure is an important thing moving forward. It is something we are exploring in our lab too. That's our next step. We're trying to add macrophages in our heart on a chip model right now. Multi-organ interactions lately being more and more explored, which will be also, I think, more and more explored in the near future. So incorporating multiple tissues and also comorbidities. So far, like we have done with aging, comorbidities in terms of like heart attack and age, like looking into differences in sex or looking into things like obesity and cancer. How another factor that is a parameter to enhance either the occurrence or the severity of the disease can be incorporated in the organ-on-chip model, and how we can leverage organ-on-chip technology to start studying multi-parameter aspects of diseases is, I think, where the field is moving and should move to.
Thank you. One final question. How do you measure the contraction of collagen gel?
It is microscopically imaged, and we have a calibrated measurement device embedded. So we have a 3D imaging system, confocal based. So we image it in 3D and look into the dimensions in three-dimensional space by measuring.
Thank you so much. That's all the questions I have. Thank you for the fantastic talk and your time, Professor Zorlutuna.
Thank you so much for the invitation, and looking forward to see you soon.
Thank you. Bye-bye.
Bye.